Welcome back to Cell Biology, everyone. We are jumping over to chapter 23 to talk about signal transduction, or how a signal on the outside of the cell can be relayed inside of the cell to cause changes in that receiving cell. So, all kinds of cells can produce signals. Unicellular organisms like bacteria can release signals to interact with other organisms. In a multicellular organism, we send different kinds of signals to be able to coordinate our cells and our tissues and our organs together to function as a multicellular organism. We do this by having different molecules on our surface that can receive signals and different signals that can be sent from a cell. One type of signaling between our cells would be endocrine signaling. When one cell releases a hormone, it travels through your bloodstream, then gets to a target cell, which is going to develop or modify its behavior in response to that hormone. Cells can also target cells next to each other that they're in contact with. That would be juxtacrine signaling. Paracrine signaling would be this cell is sending a signal. It's not physically in contact with this cell, but it's pretty close. That's paracrine signaling or nearby signaling. And they can also do what's called autocrine signaling, where this cell sends a message that is picked up by these receptors on itself. So the cell that's sending the message is the same cell that's picking up the message. It's changing its own behavior. Through these different types of signaling, we are able to coordinate our cells in our body to have functional tissues and functional organisms. When a message is left or is sent from one cell, it can bind to a receptor on its target cell. We often call this signal, this hormone or this growth factor, it's called a ligand. So the ligand binds to the receptor. It could bind to something on the surface of the cell. It could bind to something inside a target cell. And it's going to fit into the binding site or binding pocket on the receptor. And that receptor is going to have that binding site made to fit specifically for that ligand. When the ligand binds, that is the first step that's going to go through this whole signaling pathway, leading to changing something within the cell. You may remember this from when we talked about the cell cycle. So here is a growth factor, the binding to this receptor. That is going to cause changes in other molecules, so like RAS and RAF are going to have other changes. Then different things are phosphorylated, dephosphorylated, resulting in transcription of genes that let that cell go through the cell cycle. So this is just an example of a signal transduction. Signal binds to the receptor, starts knocking down dominoes. All these things result in, finally at the end, your genetic changes. These intermediates here are called second messengers. So it's not the first target, but it's signaling pathways, like the molecules involved in the pathway. Those are second messengers. And this ability to respond to this growth factor or hormone or whatever signal, this whole pathway is called a signal transduction pathway, resulting in, at the end here for this one, for growth factor, turning on genes like E2F, cyclin, CDK, to let that cell progress through the cell cycle. When the ligand binds to the receptor, there's two main ways that it can affect that receptor. One is by changing the shape, a conformational change in the receptor. That conformational change is then going to be noticed on the cytosolic side of that protein, which then results in a signal transduction cascade. Or it could cause receptor clustering. That's what we see in this image. These receptors, when they bind to its ligand, they start to cluster together. And that is going to result in the signaling cascade going on down from there. When that receptor binds to its ligand, it's going to initiate a pre-programmed sequence of events. So your cell is not going to suddenly have the ligand binding and then be like, oh, what do I do? And trying to figure out then. It has in advance planned out, has all the molecules it needs to be able to carry on its functions. And it just has those functions sitting dormantly, unused, until that ligand binds the receptor. The specific pre-programmed responses of a cell are going to depend on that developmental history of that specific cell. In a lot of cases, binding a receptor and a ligand are going to be very similar to an enzyme binding to its substrate. You can see the ligand similar to a substrate and the receptor similar to an enzyme. And when the ligand and receptor are bound, you say that the receptor is occupied. As you have more ligand available, an increasing concentration of ligand, you will have an increasing level of your receptors being occupied. And eventually that is going to get to a level that's called saturation. At saturation, most of your ligand and your receptors are going to be bound to each other. You'll have very few receptors that are unbound. You can measure how many of your receptors are occupied or unbound to see your receptor affinity. So the dissociation constant, or this KD, tells you what concentration of ligand you need to have half of your receptors occupied. So if your ligand and your receptor bind to each other really strongly, it's going to take a lower level of ligand for your receptors to be bound, because even with a little bit, they are very happy to immediately bind to the receptor, and the receptors are very happy to immediately grab those ligands. If there's a lower affinity between your receptor and your ligand, it's going to take an increasing concentration before they bind really stably. So that means that receptors with a high ligand affinity have a low dissociation constant, a low KD, and receptors with a low ligand affinity are going to have a high KD. It's going to take a higher concentration to get half of them occupied. We can make synthetic ligands, or drugs, that can bind to those receptors even more strongly than the ligand itself. And there's two different classes of drugs here that we're going to refer to: the agonists and the antagonists. So the agonists, when they bind, are going to bind to this receptor even in the absence of your ligand, presence of ligand, it doesn't matter. The agonist will bind to the receptor and turn on the signaling cascade. So you'll have the full activation of whatever genes, whatever properties would have been triggered by that cell binding to its ligand. An antagonist is going to bind to the receptor and block it. So even if you have a large concentration of ligand available, it won't be able to bind. It'll be blocked because the antagonist binds more selectively than the ligand itself. And when that antagonist is bound, you're not getting that signal. That little bitty blot is telling you you're not getting a good signal transduction being relayed. So it prevents that receptor from activating the signal transduction cascade. Then eventually you're going to need to shut down that receptor pathway. You don't want all cells constantly signaling everything at the time. You need to be able to modify and control it. There's two ways that we can shut down signaling. One is by reducing the amount of free ligand. So we just reuptake that ligand, bring it back into cytosis into a cell so it's not available to be binding to the receptor. The second is by changing the receptor. We either are going to make it less sensitive, doing something like phosphorylating it so it is less sensitive, or changing how much receptor is there, doing something like receptor-mediated endocytosis to bring those receptors into the cell so they are no longer on the surface of the cell, so they cannot bind to the ligand. Cells are also geared to sense changes in ligand concentration. That's kind of important for our cells to be able to do. So even if you have ligand available, your cell can tell that the concentration went up or the concentration went down and respond appropriately to that. So I want you to think, if you have ever walked into a room that smells bad, or maybe your house has a weird smell to it. Eventually, you just kind of stop smelling it. And then a visitor comes to your house and suddenly they're like, oh man, your house is kind of musty, or your house smells like cat or something. You stopped smelling it. You became desensitized to it because those receptors in your nose, even though they were still having that signal there, they just stopped responding to the signal of that smell. It's not just our noses that respond like that. All kinds of receptors in our body can respond to that. So when our receptors are bound to their ligands for a long time, your cell is just going to get used to it, say this is just normal now, and stop responding. So that is what receptor desensitization is. You can become desensitized to smells. You can become desensitized to drugs. We hear about that with pain meds a lot. Your cells can actually change the density of their receptors, take them off their cell surface through receptor-mediated endocytosis. And we can change the receptor's affinity for the ligand through something biochemical, most commonly phosphorylating or dephosphorylating that receptor to change its shape and change its affinity for the ligand. Another thing that's kind of cool is that your cell can take just one tiny amount of ligand and turn it into a big response. So you can have just one little hormone that then triggers your cell to respond by thousands or hundreds of thousands of effector molecules being produced. When we have a receptor binding to its target, here this would be HIV, a virus that causes AIDS, that is kind of co-opted a receptor on our T cells. And you can see this T cell receptor, the CD4 receptor, has co-receptors by it. Those co-receptors help stabilize that ligand receptor binding. And so that can help make that signal be stronger. And then as we go down through the cascade of signaling events, this receptor will turn on something, which turns on another thing, which turns on another thing. Each step is able to multiply how many molecules are being produced. So after several steps, you have, thousands or tens of thousands or millions of times more products being created. That's signal amplification, multiplying the effect of your signal through different steps. One example would be glycogen in the liver. We store glycogen in our liver cells, so we have a source of energy when we need it. One cell that can bind to those liver cells is a hormone epinephrine, which is involved in your fight or flight response. So if you are suddenly have the need to run from a bear, that epinephrine is going to kick in, release glycogen in your liver, so you have sugar molecules available to fuel your muscles to run from that bear. So when your epinephrine binds to the receptor, one molecule of epinephrine, one receptor activated. Then we're going to activate a protein attached to that receptor. And suddenly you have 10 to the second power molecules. Then you're going to activate a second messenger, 10 to the fourth molecules. Then another messenger, 10 to the sixth molecules. And eventually your response, 10 to the eighth molecules. We're going to come back and talk about G-protein coupled receptors, cyclic AMP, protein kinase. These are things we're going to talk about in a little bit. But for now, if you just know, target binds to the receptor, and very quickly through all these different signals, you have a big increase in the number of molecules being activated. So that one epinephrine molecule binding to one receptor has made your cell respond by releasing hundreds of millions of glucose molecules. So you've got plenty of energy for your body to run from that bear. Okay, cell-to-cell signals can go through some different receptors and different signal transduction pathways. There are a limited number of basic signaling pathways that we use. Most of our ligands are going to be hydrophilic compounds. They would be things like proteins, small peptides, amino acid derivatives, nucleotides, nucleosides. They're all able to, because they're hydrophilic, go through your extracellular fluid, your interstitial fluid, and bind to the receptor protein. Hydrophobic ligands can act on receptors in the cytosol, and they can move into the nucleus to regulate transcription. Hydrophilic ligands can bind to transmembrane receptors. One example would be those ligand-gated ion channels that we talked about. When the ligand binds, it opens a channel so that the ion is able to go through. This system relies on hydrolysis of GTP. So when GTP is used as the energy source, we are using G protein coupled receptors. Other receptors can involve protein kinases. So they are receptors that activate some sort of intracellular cytosolic enzyme that can phosphorylate things. And hydrophobic ligands can actually diffuse through the cell membrane and bind to a receptor within the cytosol. And then the receptor bound to the ligand goes through the cell into the nucleus and can turn on specific genes. These would be things like steroid hormones and retinoids. Okay, so that was just kind of a general introduction to signal transduction. We're going to come back and talk a lot more about G protein coupled receptors. We're going to talk about cyclic AMP. We're going to talk about kinases and phosphatases and calcium and all kinds of different ways that our cell is able to signal and coordinate with each other. I will see you soon for our next video. Bye.